Hi guys, welcome back. We are beginning part 3 for module 6, the cell. We're on page 173, the cytoskeleton. Ready? Here we go. The cytoskeleton. Before we leave our discussion of cellular organelles, we need to mention the cytoskeleton. This is not a single organelle. Instead, it is a network of fibers that holds the cell together, helps the cell keep its shape, and even aids in movement. Cytoskeleton, a network of fibers that holds the cell together, helps the cell to keep its shape, and aids in movement. The cytoskeleton is made up of 3 basic kinds of fibers: microfilaments, intermediate filaments, and microtubules. You already learned about microtubules when we discussed the centrioles, but the other 2 types of filaments are new to you. Microfilaments, fine thread-like proteins found in the cell's cytoskeleton. Intermediate filaments, thread-like proteins in the cell's cytoskeleton that are roughly twice as thick as microfilaments. Each of these types of fibers makes its own contribution to the cytoskeleton. The microfilaments are mostly associated with movement. For example, microfilaments are responsible for the ponderous motion exhibited by amoeba. They also can cause certain cells to contract. Muscle cells, for example, do their job by contracting and relaxing. The microfilaments in the muscle cells take care of this function. Finally, in some cells, the microfilaments also generate cytoplasmic streaming. Microtubules also participate in cell movement. As you've already learned, they are used to form cilia and flagella in the cells that have those structures. They also provide a track upon which organelles and vesicles can travel as they move throughout the cell. The intermediate filaments are mostly responsible for strengthening and supporting the cell, which allows it to keep its shape. Another important role of the cytoskeleton is to keep the various organelles of the cell in their proper positions. Cellular organelles are not free to float around the cell because the internal cellular structure is just too complex to allow for that. Thus, the cytoskeleton holds this complex structure together by keeping each organelle in its proper position. The makeup of an individual cell's cytoskeleton is characteristic of the task for which the cell has been designed. For example, skin cells must be very rigid. As a result, the cytoskeleton of a skin cell contains a lot of intermediate filaments so that it can hold its shape well. A muscle does a lot of contracting and relaxing, so it has a lot of microfilaments in its cytoskeleton. A paramecium needs lots of cilia, so it has a high concentration of microtubules. Although you might not think about it, there are cells in your body that have cilia as well. The bronchial tubes that bring air to your lungs are lined with cells that have cilia. These cilia filter dust and debris from the air that you breathe and then beat back and forth to sweep the dust and debris away from your lungs. As if this isn't already complicated enough, now that we know that we've just thrown a lot of information at you. Unfortunately, we have to do this because the cell is just so complex. That's why we say you need 3 weeks for this module. There's just a lot of information to absorb. So before we move on, however, we need to let you know something about Figure 6.1. In order to make it easy to see the organelles that we wanted to discuss, we radically simplified the structure of the plant and animal cells in that figure. For example, the cells are drawn as flat, even though they are not. Cells and the organelles inside are three-dimensional, having length, width, and height. Also, whereas the cells in Figure 6.1 contain at most a few of each organelle, there are in reality several of each organelle, except the nucleus, present in most cells. To give you an idea of the complexity of a cell then, the figure below shows you a less simplified drawing of an animal cell. To give you an idea of how complex cell structure can be, perform the following experiment if you have a microscope. How substances travel in and out of cells. As we learned in the previous section, the plasma membrane is responsible for substances moving in and out of a cell. If a substance is to leave the cell or enter it, it must first pass through the plasma membrane. To see how the plasma membrane works, however, we first need to see how it is made. A plasma membrane is usually constructed of proteins, cholesterol, and phospholipids. Phospholipid, a lipid in which one of the fatty acid molecules has been replaced by a molecule that contains a phosphate group. Remember from module number 5 that lipids are molecules that contain 3 fatty acids linked together on a glycerol molecule. Well, a phospholipid has, instead of the third fatty acid molecule, a small molecule that contains a phosphate group, which is composed of phosphorus, hydrogen, and oxygen. The figure below shows the structural formulas for a lipid and a phospholipid so that you can really see the difference. The bottom part of the figure also shows you the typical symbols used for lipids and phospholipids in biological drawings. The interesting thing about a phospholipid is that the phosphate group gives the molecule a slight affinity to water. It turns out, however, that since the 2 fatty acid components are so long, and since the molecule containing the phosphate group is so short, the affinity to water exists only on one end of the molecule, the end that contains the glycerol. The other end of the molecule still has no affinity for water. Thus, we say that a phospholipid has hydrophilic end. Hydrophilic means water-loving. And a hydrophobic end. Hydrophobic means water-fearing. In the little symbol used for a phospholipid then, the top part of the molecule, the end with the circle, is attracted to water, while the bottom part at the end of the wavy lines is repelled by water. Now that you understand what a phospholipid looks like, you can learn how a plasma membrane is constructed. A plasma membrane contains proteins, cholesterol, phospholipids, and other molecules arranged as shown below. In the plasma membrane, phospholipids are arranged in a double layer called the phospholipid bilayer, with the hydrophobic ends pointing towards each other and the hydrophilic ends pointing out towards the surroundings and in towards the cell. This should make sense because water is both outside the cell and the surroundings and inside the cell and the cytoplasm. Thus, the hydrophilic ends will point towards the water and the hydrophobic ends will point away from the water. Proteins and cholesterol are interspersed throughout the phospholipid bilayer. Some proteins are near the top of the bilayer, and others are near the bottom. In addition, some proteins span the entire width of the plasma membrane, leaving no room for phospholipids. This protein bridge can be an active transport site, which we will discuss in a moment. In addition to what we have already discussed, there are carbohydrate molecules attached to the outside of the plasma membrane. When a carbohydrate is attached to a protein, the protein is called a glycoprotein. And when it is attached to a phospholipid, the result is a glycolipid. The prefix glyco refers to glucose, which you should recall is a monosaccharide that can link together to form large carbohydrate molecules. The amount and type of carbohydrates on the plasma membrane varies from one cell type to another, as well as from one individual to another. As a result, they serve as a means of cell-to-cell recognition. For example, people have 4 blood types: A, B, AB, and O. These blood types are distinguished from one another by the arrangement of carbohydrates on the plasma membrane of a person's red blood cells. One neat feature of the plasma membrane is that it can self-reassemble when it is broken. When a secretion vesicle, for example, reaches the plasma membrane, the membrane actually breaks to allow for the release of the secretion vesicle's contents. Afterwards, the plasma membrane reassembles by itself. This self-reassembly is due primarily to the fact that the hydrophobic ends of the phospholipids are attracted to one another, and the hydrophilic ends are attracted to the water on the inside and outside of the cell. Thus, the phospholipids always end up pointing in the right direction once they have time to regroup. There are 2 basic ways that substances enter or exit a cell: passive transport or active transport. Passive transport: movement of molecules through the plasma membrane according to the dictates of osmosis or diffusion. Active transport: movement of molecules through the plasma membrane, typically opposite the dictates of osmosis or diffusion, aided by a process that requires energy. Now, remember, you have already learned about osmosis and diffusion. If you have forgotten what they are, you should go back to module number 5 and review those concepts. In passive transport, no cellular energy is expended as a result of the substance passing through the plasma membrane. Water, for example, is a very small molecule and thus passes easily through the spaces between the proteins in the plasma membrane. As a result, it will travel according to the dictates of osmosis. Thus, if the concentration of solutes in the solution surrounding the cell is essentially equal to the concentration of solutes inside the cell, water will aimlessly wander back and forth across the plasma membrane, resulting in no net change in the amount of water inside or outside the cell. When cells are in a solution whose solutes are essentially equal in concentration to those of the inside of the cell is said to be in an isotonic solution. Isotonic solution. A solution in which the concentration of solutes is essentially equal to that of the cell which resides in the solution. The red blood cells in your bloodstream, for example, are in an isotonic solution. Your kidneys get rid of excess solutes in the blood to make sure that the bloodstream stays isotonic with your red blood cells. Why do your kidneys need to make sure your bloodstream stays isotonic with your red blood cells? Well, remember the story about the rhyme of the ancient mariner from module number 5? If cells are placed in a solution that has a significantly higher concentration of solutes than that of the cell, osmosis will drive water out of the cell and into the surroundings. This will cause the cell to lose too much water, killing it. Solutions that have a higher concentration of solute than the inside of a cell are called hypertonic solutions. Although a hypertonic solution has the potential to kill any cell, the implosion that results from placing cells that have cell walls in a hypertonic solution is given a specific name: plasmolysis. Hypertonic solution. A solution in which the concentration of solutes is greater than that of the cell that resides in the solution. Plasmolysis. Collapse of a walled cell's cytoplasm due to a lack of water. This is why you should not over-fertilize a plant. If you put too much fertilizer in the ground near a plant, when the fertilizer mixes with the water in the soil, a hypertonic solution could be formed. This will result in plasmolysis of the plant cells, killing the very plant that you were trying to fertilize. Osmosis can also be called cytolysis, when a cell is placed in a hypertonic solution. Cytolysis, the rupturing of a cell due to excess internal pressure. Hypotonic solution, a solution in which the concentration of solutes is less than that of the cell that resides in the solution. In a hypotonic solution, there is greater solute concentration on the inside of the cell than on the outside. As a result, water flows by osmosis into the cell until the cell bursts from too much water pressure. Since many organisms live in freshwater habitats, a lot of cells must deal with living in hypotonic solutions. After all, fresh water has few solutes dissolved in it. Thus, it is a hypotonic solution for cells. Plant cells typically deal with living in a hypotonic solution by using their central vacuole and their cell wall. As discussed in the previous section, as water flows into a plant cell, it collects the central vacuole. This expands the vacuole, pushing the cytoplasm against the rigid cell wall. This results in an increase in pressure, which we called turgor pressure. Once the pressure gets large enough, it counteracts the effect of osmosis, and no more water can get into the cell. Even though the water wants to get into the cell due to osmosis, the turgor pressure keeps the water out. This is what we mean when we say that turgor pressure counteracts the effects of osmosis, keeping the cell from rupturing. Plant cells can do this, of course, because they have a rigid cell wall against which the cytoplasm can push, causing turgor pressure. What about cells that must live in freshwater environments but do not have cell walls? Well, if you think back to module number 3, you will remember that certain protozoa have contractile vacuoles that constantly pump excess water out of the cell. Animal cells that live in hypotonic solutions also have those vacuoles. Thus, they cannot stop water from entering the cell as plant cells can. But they do get rid of it right away, so excess pressure does not build up. Actually, when scientists first observed contractile vacuoles, they thought that they were looking at hearts because the contractile vacuoles pumped constantly, just like hearts. What they were doing, however, was just forcing water to leave the cell. When the contractile vacuoles force water out of the cell, as discussed above, they are engaging in active transport because the cell is expending energy to cause a substance, water, to travel across the plasma membrane against the dictates of osmosis. Another example of active transport involves the proteins we discussed earlier. When a protein spans the width of the plasma membrane, it often has been fashioned to recognize a certain type of molecule. When it recognizes that molecule, it goes through a series of shape changes that pulls the molecule across the plasma membrane and into, or out of, the cell. That's why the area pointed out in figure 6.7 is called an active transport site, because the protein actively works to transport molecules with the help of energy provided by the cell. An example of this can be found in potassium. Many cells take in potassium, but the concentration of potassium in a cell's surroundings is usually low compared to inside the cell itself. As a result, the cell must expend energy to pull potassium across a protein in the plasma membrane and into the cell. If you have a microscope, perform the following experiment before you go on to the next section. It will give you the opportunity to see some of the processes that we have been discussing. How cells get their energy. Cells produce usable energy in a chemical reaction that, at first glance, seems rather simple. This chemical equation represents what happens when a molecule of glucose is burned. 6 molecules of oxygen from the air interact with the glucose molecule. The atoms rearrange, forming 6 carbon dioxide molecules and 6 water molecules. As is the case when anything burns, energy is also released. This is, in essence, how a cell produces the energy that it uses to sustain all of the processes it must run. Of course, we say in essence because the actual process of burning monosaccharides in cells is actually quite complicated. Although you've probably never tried it and you really shouldn't, it makes a mess. Sugar does burn. If you strike a match and light some sugar, the sugar will burn, producing water, carbon dioxide, heat, and smoke. The heat is a form of energy. Although you expended some energy striking the match, once you get enough sugar burning, the heat that it generates will be much, much more than the energy you spent using the match to light the sugar. You see, the chemical reaction described by the equation above needs a bit of a push to get it started. This push, called activation energy, is supplied by the match. Activation energy. Energy necessary to get a chemical reaction going. Once the chemical reaction starts, however, it produces lots of energy. Some will be used as activation energy to start more reactions and thus keep the fire going, while the rest will be used to produce heat and light in the form of flames. Now, it turns out that all chemical reactions require some amount of activation energy in order to start. Reactions, such as the burning glucose, however, end up producing a lot more energy than that which is required to start them. The net result is lots of energy that can be used for other things. If a mitochondrion were simply to burn sugar in the way that we just described, however, the results would be disastrous. The reaction of glucose and oxygen is very fast once it gets going. It produces a large amount of energy in a short amount of time. This large concentration of energy in a short time interval would be so violent that it would destroy the mitochondrion. So the cell simply slows the reaction down, right? Wrong! You see, the cell needs a lot of energy. If the cell were to slow the burning of glucose down to a point where the mitochondria could withstand the heat, there would not be enough energy produced to allow the cell to run all of the processes that it needs to run. Thus, something else must happen. What happens is really quite complicated. We will give you a brief introduction to it here, but please realize that the actual processes involved are quite a bit more complicated than anything that we will do here. This is simply a short, sketchy introduction into one of the most complicated topics in biology: cellular respiration. Cellular respiration can occur aerobically or anaerobically. In this course, we will concentrate on aerobic respiration. Aerobic respiration is accomplished through a four-stage process that begins in the cytoplasm. When the cell wants to burn a monosaccharide for energy, the monosaccharide first goes into the cytoplasm. There, enzymes catalyze a reaction that causes the monosaccharide molecule to lose some of its hydrogen atoms and form two molecules of a substance called pyruvic acid. A bare bones chemical equation that describes this process is glucose and pyruvic acid plus 4 hydrogen molecules plus energy. The cell has to provide the activation energy required to get this to happen, but that's okay. Much like striking a match to start something burning, the amount of energy needed to begin this reaction is half of that which is released when the reaction finishes. This is the first stage of cellular respiration and is called glycolysis. Once two molecules of pyruvic acid are made, they head to a mitochondrion. As they cross into the mitochondrion, the next stage, called formation of acetyl coenzyme A, takes place. This stage is also called the oxidation of pyruvic acid in some biology texts. In this process, the pyruvic acid is broken down and a protein, coenzyme A, is attached to the remains. A bare bones chemical equation for this process can be written as follows. Now, don't get put off by the equation. It's not that bad. It just takes the two molecules of pyruvic acid produced in glycolysis and attaches two molecules of coenzyme A to them. Since coenzyme A is a big molecule, we do not give its chemical formula. We just call it by its name in the equation. Now, notice what happens in the reaction. Pyruvic acid has 3 carbons in its chemical formula. When the coenzyme A reacts with the pyruvic acid, a carbon is removed. You can see this by looking at the chemical formula of the first product, acetyl coenzyme A. It has 2 carbons in its chemical formula instead of the 3 that pyruvic acid has. Where did the carbon go? It went into the second product, CO2, carbon dioxide. The pyruvic acid also lost a hydrogen atom and 2 oxygen atoms. The oxygen atoms are used to make the CO2, and the hydrogen atom is the final product in the equation. Well, friends, let's pause again. I will see you next time for part 4.